Hello, how are you? So dear students, today we are going to take plant physiology. Plant physiology basically, it is the, you can say, branch of biology. So it can be animal physiology, it can be plant physiology. In plant physiology, we are dealing with the functionality of a cell, the vitality of a cell, how cell functions with its different surroundings, with water, with solute, with different minerals, and that's what is physiology. So it's basically study of a living state or functioning or vitality of a cell. So we are taking the first section of this plant physiology, and that's transport in plants. Okay, so let me start the thing. So this is plant physiology. Fine. So this is plant physiology. Now the first important chapter, I would guess, in the topic is transport in plants. We can write transport in plants. So this is what is our topic in today's lecture. So transport in plants, basically the plant is a living organism, so it has to grow, it has to make use of water, it has to take minerals, it has to take different solutes, and it has to take phytohormones or plant hormones or growth regulators from one part of a plant to another part. So basically, in order to move different chemicals from one part of plant to another plant, it needs kind of a transport. It needs kind of a translocation. So like in animals, there is a vascular system, which is basically blood, heart and vessels. In plants, we have this system which is used to transport different chemicals from one point to another point. If you see plant, if you see cell, the cell is very important. The plant cell, so basically if this is a plant cell, now this plant cell, it has got the nucleus and vacuoles. It also has this what's called the cell wall. So this is cell wall. So this is cell wall. This is plasma lemma, plasma membrane, plasma lemma, and this is vacuole. This is nucleus. So this cell, it needs water, it needs minerals. So there the water can be taken inside cell, or it can be released from cell to outside, to neighboring cell, to neighboring environment. There is also movement within cytoplasm, and this water needs to be transported from one cell to another cell, or from roots. For example, this is soil and this is plant root. Water has to be shifted from here to apex, to leaves, to different floral, or we can say to floral parts, or leaf parts, or shoot parts. So the transport can be, definitely it can be long transport, or it can be short transport from one cell to another cell, or from cell to neighboring environment, whatever that is. Plus there is a movement within cell. So transport in plants, basically we can broadly classify it into three categories based on distances. So we can have a cytoplasmic transport, which is a very short distance transport within cell, cytoplasmic streaming. So we can say cytoplasmic streaming. Cytoplasmic streaming is basically movement within cytoplasm, within protoplasm, within this single cell. This first category. Second category is short distance transport in plants. Short distance transport. The short distance transport is, for example, from one cell to another cell within same tissue. So it is a very short distance in centimeters even millimeters, or even we can say some inches, or even up to feet, so are less than that, that is a short distance transport. And the third one is, which is called long distance transport. So this is long distance transport. So as you remember, as I said, that the molecule water molecule or a solute molecule or mineral or whatever it is, it can move from this part of a cell to this part within cell, which is called a cytoplasmic streaming. In second case, it can move out of cell to the neighboring cell or to another cell, but within same tissue. This is short distance transport. Then we have long distance transport. For example, the xylem transports water from roots up to various parts of plant. Likewise, phloem, it transports solute from leaves to different parts of plant. So basically we have three categories. This long distance transport is also called translocation. It is called translocation. This is what is called translocation of solute. So translocation is basically movement of a particular chemical or solute from one part to another part, which have some kind of distance between them. So these three categories are basically three broad categories of transport in plants. Now, if we see the transport of water or xylem-based transport, that is unidirectional. But the transport of solutes, or which is called translocation from leaves to different parts through phloem, is bidirectional transport. So we have, based on direction, we have two kinds of transports. For example, this is plant. Now water moves from this root to different parts. So through xylem, water moves. H2 is water, it moves. This is called, what I said, unidirectional transport. So this is unidirectional transport. Fine. Because the water moves from roots to different parts, but in a one single direction. So this is direction. But in case of, or we can say direction basically is in a single go. The water moves in a single go. So water, once it starts moving from roots, it moves to different parts in a single go. But in case of phloem, in case of solutes, there is a bidirectional transport. Bidirectional transport means the transport occurs twice. Bidirectional transport means the transport occurs twice. That means if this is leaf, it has synthesized some kind of a solute. Now this is plant. The first transport of solute from this leaf, the solute moves from leaves to different storage organs. So this is storage organ. Suppose this is storage organ. The solute moves from leaf, from synthesizing organ to storing storage organ. This is first transport. This is first go. In second go, the solute gets distributed from the storage organ to different parts of plant where it is used. So this is first transport and this is second transport. So the two transports happen in case of solute, but in case of water, which is carried through xylem, there is a one single directional transport. So this we have two directional transport. Now let's move, because this topic deals mostly with short distance transport. So if we take short distance transports, we have two important types of short distance transport, as I write here, which is very important. And you can also keep your rough and pen handy in order to write this before writing it out. So right now, let me start here. Transport in plants. So basically we can say is long distance transport. Long distance transport. Then we have which is called short distance transport. Short distance transport. Okay, so we have two types of transport in plants. So first is short distance transport. The second case, which is called a long distance transport, is what exactly I said, is called translocation. Translocation. Okay, so this is what we are dealing with: solute, translocation of solute through phloem. That's the next topic coming in series. Now this is short distance transport, this is long distance transport. The short distance transport basically is of two types. It is basically of two types. We have, which is called passive transport, and active transport. So this is active transport, this is passive transport. The difference is that in case of active transport, we need to have there is expenditure of energy, and in living organisms the energy comes in the form of what is called adenosine triphosphate, that is ATP is used here. In active transport we require the use of ATP, but in passive transport no expenditure of energy occurs. So this is the main difference between two. Now this passive transport, it is of two types. It is of further two types, which first is called diffusion. So this is called diffusion. Second is called facilitated diffusion. Facilitated diffusion. So here the diffusion occurs only through facilitation. Now what is diffusion and what is facilitated diffusion? Then we have to take this active transport. So in case of active transport we need ATP expenditure of energy. In case of passive transport no expenditure of energy occurs. Now this passive transport, where there is no requirement of energy, we have two types. First is diffusion, second is facilitated diffusion. Now let us try to understand this concept of diffusion. Do you know diffusion, as we have studied in chemistry or physics? The diffusion is just a movement of molecule or a chemical from its higher concentration to its lower concentration, and that's what diffusion is. For example, this is glass of water. In this glass of water, it is filled up to this point, it is filled with water. Now if we drop ink, a drop of ink here, it does not remain confined to this space. It moves from here in this way till whole water gets colored with that particular ink. So that means the ink drop of water, it moves from higher concentration to lower concentration, where there was deficit, where there was no ink particle. Likewise, if we put kind of a sugar or solute or salt at the bottom of this glass, at 0 point of time it will be separated and there will be no solute inside this area of water. But with time this starts diffusing, the diffusion starts occurring. The solute goes on to make this whole water as solution. So it was solvent and this was solute. It becomes finally what's called solution. So the solution comes up. So basically diffusion is a transport, or it is movement, you can say movement of a molecule. Suppose these are molecules and this is medium where there are no molecules of this particular type. Suppose this is X molecule. We can say that diffusion is movement of molecules from their higher concentration towards from the area of their higher concentration towards area of lower concentration. So definitely this movement occurs across concentration gradient. So there is a concentration gradient. That means this is low concentration, then high concentration, then high concentration. When we reach here, we have the highest concentration. That's what exactly we can study in graph. Suppose this is 0 concentration, This is full concentration 100%, this is zero concentration. With passage of time, the concentration of this particular molecule, we can say it decreases from here to here. So concentration goes down. When we reach this point, we will find zero concentration. But with some point with passage of time, the concentration graph becomes like this. It is equal. So the concentration gradient is formed basically, and this movement of this particular chemical, it goes across concentration gradient. So concentration gradient is basically a gradient where we will follow the path of higher concentration to this path of this up to the point where there is zero concentration. So this is a gradient. The movement is across gradient like this till equilibrium reaches. Till there is a equilibrium. So that means both areas have got same concentration of a particular chemical. So now time reaches. You will find that throughout this path the chemicals are equal. The chemical is distributed equally. Now there is no movement. Even if there is a movement from here to there, another movement will cancel this movement. Suppose the molecule moves now here to there, another molecule moves from here to there. The net movement can be set to as zero. So net movement is zero because if one molecule moves here, other molecule cancels that moment. It is motion. Another molecule moves from here to there. This point, which is called moment of net zero moment, this is what is stage which is called equilibrium stage. So before reaching equilibrium stage, there is a continuous movement of molecule from higher concentration to lower concentration. So basically what is this? Diffusion is diffusing of a particle from higher concentration to lower concentration. Okay, clear? Now if we take example in cell, for example in case of living cells, that's what exactly we are dealing with. Plant cells. Suppose this is plant cell. Now what will happen if this is plant cell? Suppose there is one molecule, for example sodium or some other ion. Now the concentration is very much here. It is 100 suppose parts per area, and inside cell it is zero. The sodium start entering inside cell and till the equilibrium reaches. That means the point where both inside and outside of cell is having equal concentration of sodium, that is equilibrium point. Then till that point the movement will continue. Suppose if there is potassium inside cell, outside cell there is a zero potential start moving outside. So basically diffusion is simplest form of transport, where there is transport of a mineral or material from higher concentration to lower concentration. And this is very important for plants because most of the cell functions depend on this diffusion. This is diffusion. Now we have facilitated diffusion. As term says, the diffusion occurs, but we have to provide some kind of facility. The important thing for facilitated diffusion is that there has to be a concentration gradient, there has to be a concentration difference. If there is no concentration difference, then it will not be called a diffusion. So basically the first criteria is concentration difference should be there. Then even if it starts moving, because of concentration gradient, the solute starts moving, but it cannot move because there are some problems. By making those problems, by clearing those problems, the diffusion occurs. So basically it is a diffusion, but we need to have kind of some facility so that the molecule can enter cell. Suppose for example, again this is cell, now this is cell membrane. Suppose this much, there are molecules. Molecule A, it wants to enter this cell, but it could not because it can be hydrophilic, water-loving molecule, but cell has lipids. The cell membrane is a hydrophobic. It is lipid-loving. We can say it is water-hating. So cell membrane is hydrophobic, it is hydrophilic. Then it cannot enter cell. So there are some proteins. There are some proteins on the cell which make this A to enter through them and enter into the cell. These proteins are very important, and these are what are called facilitators. These are facilitators for facilitated diffusion. This is very important. Now these proteins, there are many kinds of proteins for different molecules. Here one important point is that the specificity starts becoming more prominent. So what I mean to say is that in case of diffusion, there is no selection. There is no selection of molecules, because any molecule having more concentration outside can enter inside cell depending on concentration gradient. The selectivity is not here in case of diffusion, but in facilitated diffusion, the starting of selectivity occurs. That means if there is a particular protein inside this protein enters only A, so only A will enter. Suppose there is another protein which enters only P, so only P will enter. So this is actually start of we can say the formability of a cell starts selectively selecting different materials. So in overall, what I try to I want to make you understand is that the point is called selectivity starts happening. So selectivity of chemicals. If we make this more, if we try to understand it more deeper, this is what is called evolution in kind of transportation in plants. So the plant life starts modifying, starting evolving more. That means adaptation comes into play, and this is what is molecular inside molecular adaptation. We can say selectivity occurs because in case of diffusion, no selectivity, it was simple diffusion. In case of facilitated diffusion, selectivity because of different proteins occurs. Now these proteins are basically called, there are some important categories, porins. So this is porin proteins. So porin proteins have a channel inside them. They make entry of a particular chemical. Or there are some water channels also, which are called aquaporins, because aqua means water. These are called aquaporins, which are basically water channels inside cell membrane. So water comes through these aquaporins. Now porin proteins are very important, and there are different classes of porins depending on the chain they have, the alpha chain, the beta chain, type of a protein, type of amino acid present in them. This is what is important. Now this diffusion and facilitated diffusion, they depend on some important criteria. If we see the criterias, those are definitely diffusion can depend upon the concentration. Important is concentration. Second is it can depend on temperature, but that's a different point to be taken later. So diffusion on one side we can see it is independent of that temperature. So diffusion is independent of temperature, but this facilitated diffusion can depend on temperature, because the porins, in case of high temperature, the porins get denatured, or these porins present in cell membrane, they get deformed, they get denatured, and their function depends on temperature. But overall this diffusion does not depend on temperature characteristics. And then there is a pH concentration, we can say. Then there are many other criteria. So this is what is called this passive transport. Now we have one more, which is called active transport. Now active transport is, because it is active, it uses energy. Why it uses energy? Because the moment of molecule is opposite to that of concentration gradient. So we need, it is an uphill task. We need to have kind of energy, because in case of diffusion, it was from area of higher concentration to the area of lower concentration. But active transport, we need to transport molecules from area of lower concentration to the area of higher concentration, which is not possible without use of energy. So energy is required because it is uphill task. We have to ride a kind of hill. It is not downhill task. Here it was downhill task, but in case of active transport, it is uphill task. We are moving molecules against concentration gradient. For example, there is a cell, plant cell is there. There are 10 molecules of sodium inside, 2 molecules of sodium outside. Now the diffusion will definitely occur in this way, but we do not need that. We need these two molecules should also go inside, but this is against the rule of diffusion. Then we need energy. Energy is used, which makes these molecules also go inside. So that means we are making transport of molecules against concentration gradient. Now what happens also in case of active transport, we have different proteins which are called permeases. So suppose this is cell. Now in this cell, if you see this is cell membrane, for example, the molecule wants to be inside, so we need to transport this A molecule towards cytoplasm. There are different proteins. So these different proteins have different binding sites. They have different mechanisms by the help of which this protein, suppose this A does not fit on this protein, then what happens? With the help of ATP molecule, which gets converted to ADP molecule plus P. The ADP plus P, that means ATP is broken down to ADP plus P. This P bond, which is having a high energy, phosphorest bond, this P bond, when this P gets separated from this ATP, there is a release of energy, because when we make ADP plus P, when it gives ATP, there is a utilization of energy. But the reverse of it is that ATP broken down to ADP plus P, energy is released. This energy is utilized in deforming or making this protein functional. It changes its shape in this way. For example, the molecule enters inside it, the cell, the protein gets again into the previous shape, or in this way. So it is the cyclic phenomena. We can write this this is protein, this is molecule to be transported inside. Now these come together with the help of ATP getting converted to ADP plus P. This protein is taken inside, okay, and this molecule is again released back. So it is a cyclic phenomena. Okay, now one more important point is that, which is actually part in case of diffusion and facilitated diffusion, we have two important, or in fact 3 important terms, which is uniport, symport and antiport. Now for example, this is cell membrane, okay, this is cell membrane. Now if only single, suppose A gets inside cell, the membrane is such it allows only single molecule to come inside. This single transport is called uniport. Now if it is like that, this molecule is such that A gets only inside when it has when it has B with it, that means A gets inside only after having B with it. So A, B both getting inside. A cannot go singly inside, B cannot go singly inside, but both can go inside when they are together present. This is called symport. Symport means they are simply supporting each other in order to enter, and the direction is, the symport is unidirectional. We can say both are coming inside or both are going outside, so there is a one direction. But in case of one more point, that's called the antiport. The molecule is such A comes inside only when B goes outside. So A comes inside, B goes outside, and when B goes inside, A goes outside. So these are reverse of each other, opposite of each other. This is called the antiport. There are a lot of examples of antiport, symport, uniport, and this is again what is called selectivity, and selecting particular molecule to be entered. And you will see in a number of examples. In case of glucose, gets only transported through some chemicals like sodium. When sodium is present, glucose goes inside, otherwise not. And even in case of Krebs cycle and photosynthesis, respiration, etc., chloroplast membrane, there are many transports, imports and antiports. Those will be taken in, the examples will be taken in chloroplast and when we are studying this mitochondria. So the simple concept is that uniport is single transport in one direction, symport is also in one direction, but there are two molecules, they should be present. In case of antiport, the transport is opposite. Okay, now this is what we called short distance transport. The long distance transport, which is called translocation, will be taken separately as a different lecture, different topic. Now we are now moving to the cell, which is called plasmolysis. Then we are doing what is called exocytosis, endocytosis, or we can say plasmolysis, deplasmolysis, different types of plasmolysis, and osmosis, in fact reverse osmosis, and water potential, pressure potential, solute potential. And these all things will be taken in different lecture. But this was basic. We are starting plant physiology. This was basic transport in plants. This is the first chapter, we can say. And the next topic will be dealing with what's called water relations. In that, we'll be dealing with osmosis and all that potentials. And I hope we will meet in next video with one more important topic. Take care. Goodbye.